Hi Hires, so we are on Metabolism and Survival. We are looking at growth conditions for microorganisms. Okay, so let's start off with why you would use microbes in the first place. And these are single-celled organisms and they are used for sources for so many different things that we use from biotechnology. And it really has come down to the fact that they are everywhere. They live in such a wide variety of conditions that they have pathways that involve a huge number of different metabolites. And that means if we can find the right one, then we can find the right thing. They are generally pretty easy to cultivate, to keep. It quite often is not very much stuff that you need, and you don't need particularly complicated culture requirements. And they grow fast. Now you know that in particular because we've had that as part of the whole speed of evolution of microbes as well. Fast generation times means that you can tweak it to get an extremely high production rate. Okay, so types of microbes that we're looking at, basically any of them. Any of the main domains of life that you know, the bacteria, the archaea and the eukarya, any microbe from there. So in the eukarya, we're generally looking over at your fungi, maybe in the plantae, you might be looking at algae and things like that in here. But we have lots in here. Any one of these, any of these, we might be able to find something useful. All examples. Okay, so the growth conditions that we might be looking at here. First off, we need to consider the energy source. So everything needs an energy source. If it's a photosynthetic microbe, so a form of algae, then that might just be light. It's more likely to be chemical, so some form of sugar is the most common one, but it might be specific for that particular microbe. So energy source is the first thing we need to do. Then we need raw materials for biosynthesis. That sounds a bit posh, but basically what we're saying is you need all the things that would normally make you produce something big. So if I'm going to make things like proteins, I need amino acids. If I'm going to make DNA, I need the building blocks and nucleotides. So all of these things are necessary in the culture. So as well as the basic energy source, you want to provide them with water, some carbon, some nitrogen, some mineral salts. All of that would be the basics, and if they have all these things, then they can make the things that would make something bigger. So raw materials for biosynthesis. On top of that, we have some things that are individual for particular microbes. So what do we do once we know what we need? Once we know what we need to suspend it all in something that they can grow in. And the most common one of these is agar. So agar is a solid medium which allows you to grow microbes on the surface of the gel. So you've done this with agar plates. Okay, you also can have liquids, which like a culture broth, which again you can just put everything you need, and that will allow them to kind of keep moving around, so they grow throughout the liquid medium, but on top of a solid. The important thing is just making sure you've tweaked it so everything you need is there. The one that you have used and will have seen most commonly, but wouldn't really necessarily have thought about what's there. Nutrient agar obviously contains agar, but it also has some polypeptides, has meat extract, some salt, and then agar. Beef extract is really common because it gives you a lot of the complex compounds that loads of them need. And then we have just a wee picture here of what a blank agar plate might look like, and one which has been colonized by quite a lot of E. coli. Right, so we've got an energy source, we've got the stuff they all need for biosynthesis. Our next thing is get rid of any competition, because competing with other microbes will slow down the growth, or worst case scenario might stop it entirely if the other one takes over. So what you want to do at the very beginning is deal with aseptic techniques. So wipe out anything else that is not the thing that we want. Aseptic techniques is just a general term for all the procedures you do to ensure that you have sterile conditions at the beginning and that you avoid contamination. Autoclaves, disinfectants, lab coats, gloves, flaming, anything which you could, all of this are just basic aseptic techniques. There's a whole pile of that in that 5 information. Fourth one we can look at, so we've got energy source, we've then got our biosynthesis materials, we've then got no competition, and then we just go for the optimum environmental conditions. Now that should not surprise you. If it's something that requires a specific temperature to be optimum, go for that temperature. Don't make them work hard, that's silly. And that's basically it. Oxygen levels are also in there, pH, that's the best one for them. Just make it as nice as it can be, and that's important for them. There is a little side note to this one. Some metabolic pathways that we want them to do might not be the ones that they do if they're in optimum conditions. You might want to actually stress them. And if that's the case, you may actually want to change one of these, but then you're still in control of what's happening. To make this actually work well, what we're looking at is a fermenter. And this is your kind of basic picture of this kind of thing. So on the left hand side we have an actual picture of a rather large fermenter. That's kind of probably about person size, and this is the kind of diagram of it. So what we've got is these would be all the probes, and it looks like we've got various things that we can feed in and out. Again, we've got substrates going in and out of here. Looks like substrates in, products coming out the bottom, and we've got a whole pile of stuff controlling pressure, and this would be the water cooling to keep it at the right temperature. So it's slightly easier looking at this one to give you a kind of better version, okay? But nothing new in terms of what we're doing here. Okay, some things that might actually be tweaks to what you're doing. So there are some microbes that you need to take your kind of basic nutrient agar or whatever you're using as your standard and dump in some extra stuff that they need. Okay, it could be things like blood. It's quite often used if we need something that only lives in blood. It could be serum, could be egg, could be things like that. We might actually use also differential media, and that means that we can use something that shows up a particular thing about what's growing there. So it might tell you that everything that comes up blue is taking in a particular dye and therefore has a particular thing we want, that kind of thing. So the other type you can have rather than differential is a selective media, and a selective media is basically doing exactly what it said. It is selecting for particular microbes. So say for example you had a mixture of microbes in there, but some of them you'd been genetically engineering, and the ones you've been genetically engineering have been engineered to have a resistance to a particular antibiotic. So you put that antibiotic in the media, and the only ones that will survive will be the ones that have that particular antibiotic resistance. Here's a very specific example. Lowenstein-Jensen media. This is for culturing Mycobacterium tuberculosis and contains a whole pile of things that basically wipes out everything else, but only the Mycobacterium then grow. So overall, the reason that we like microbes for biotech are they're adaptable. The fact that their adaptability gives you a range of products, and it's an easy thing to culture. You can control the energy source, everything else they need, the environmental conditions, and competition. If you do all that, you will get a lot of growth from your microorganism. That's it.